Anaerobic culture for oat surface sample. Purpose: the regular monitoring of pathogens in the oats is one of the preventive measures for postoperative infections. Clostridium tetani is one of the major threatening anaerobic bacteria of oats, as clostridial spores easily spread into the air. Principle: regular monitoring of oats for screening of clostridial spore by inoculating in thioglycolate broth for anaerobic culture should be performed. This broth supports the growth of anaerobic and fastidious microorganisms. Microbiological surveillance of oat by surface swab method for anaerobic infection and identify the clostridial spores, Clostridium tetani. Type of container: sterile cotton swabs. Equipment: sterile cotton swab, laminar airflow, incubator, gasbox, anaerobic jar. Reagents: thioglycolate broth, spore stains kit. Anaerobic culture procedure: sterile swabs moistened with sterile distilled water and rub the surface of the different sites. Inoculate the swabs into a tube of thioglycolate broth and incubate at 37 degrees Celsius for 72 hours in gasbox anaerobic jar. After incubation, the results will be reported as the presence or absence of Clostridium spores after endospore staining, Schaefer-Fulton method. A. Add malachite green stain solution and steam for 5 minutes, keeping the paper moist and adding more dye as required. Alternatively, the slide may be steamed over a container of boiling water. B. Wash the slide in tap water. C. Counterstain with 0.5% safranine for 30 seconds. D. Examine the slide under a microscope for the presence of endospores. Endospores are bright green and vegetative cells are brownish red to pink. Examples of positive endospore staining: Clostridium perfringens, Clostridium botulinum, Clostridium tetani, Bacillus anthracis, Bacillus cereus, Decylphotomaculum species, Sporolactobacillus species. Interpretation: if presence of turbidity, make a smear and perform endospore staining. Look especially for organisms morphologically suggestive of Clostridia, Clostridium tetani. In the absence of detection of bacilli suggestive of Clostridium tetani, hold culture for 7 days and report as negative if no turbidity. Reporting of test results: absence of Clostridium spores, only brownish red to pink vegetative cells were seen. Presence of Clostridium spores, green endospores located in the terminus, drumstick appearance, of the cells were observed, suggestive of Clostridium tetani.